Now we'll have the next speaker, and that is Ian Hickson from University of Copenhagen.
Thank you, Lena. And I'd like to thank the organisers for giving me the opportunity to speak at this conference. My lab, and indeed the centre I'm part of, focuses on genome instability and how genome instability impacts on age-related human disease. We have interests in cancer, interests in neurodegeneration, and in, for example, ovarian aging. I'm going to be talking about a quite famous family of proteins in human cells which have been associated with the genome stability field, but also associated with aging and age-related diseases, and that's the RECQ helicase family. I'll be talking about one member of that family, which is the BLM protein, which is defective in a disease called Bloom syndrome. And then I'll be talking about how the BLM protein is involved in suppressing aneuploidy. So I'll be talking about the same topic that you heard about from Björn. So this is this family of proteins, the RECQ helicase family. It's one of the most highly conserved families of proteins in evolution, named after a bacterial protein. It's conserved in budding yeast with SGS1, and then there are 5 family members in human cells. And one of the main interests really in this family of proteins, other than the fact that they're essential for the maintenance of genome stability, has been the fact that they are mutated in recognizable human diseases. Now, I have to introduce proteins that bind to BLM because they're of relevance to the later in my talk. And this is the so-called TRR complex, which is topoisomerase 3, RMI1 and RMI2. So I'll just refer to that as TRR. Now, I mentioned that this family is associated with human disease, and 4 family members now in humans have been identified in recognizable disorders. And I'll go through those in turn. The first one is Bloom syndrome, which is mutated in BLM. That's our kind of favorite protein, you might say. It's associated with a whole series of obvious abnormalities, mainly dwarfism, proportional dwarfism. The individuals often have a rather progeroid facial features. They have sunlight sensitivity, compromised fertility. So, for example, males are infertile. Females are actually fertile, but they often enter the menopause in their 20s. And the reason why this disorder has been studied so extensively is that it's been associated with cancers of all types. As far as I'm aware, it's the only cancer predisposition syndrome that's known, where every type of cancer is seen that you get in the normal population. It's just seen very early in life. And the chromosomes from these individuals are highly unstable. The other RECQ helicase disorders I'll just quickly go through. I mean, this is the famous Werner patient who is shown all around the world, who was phenotypically normal at the age of 15 and then showed very obvious multi-organ premature aging only in her 40s. Then there's RECQ4 deficiency diseases. There are actually 3 different diseases that are mutated in this protein called RECQ4. And those diseases are mainly associated with skin and skeletal abnormalities. And then very recently, in the last year, in fact, there's been a new disorder that's been identified that's mutated with the RECQ1 protein, and that's called Recon syndrome. This is also associated with primordial dwarfism, with a rather progeroid face, for some reason a hyperplastic nose, and then some abnormalities that are more characteristic of other diseases like xeroderma pigmentosum, where you get xeroderma and skin photosensitivity. But if we get back to BLM and its complex, I mentioned to you that it had these partner proteins called the TRR complex. Well, actually, recently, again, mutations have been found in individuals with primordial dwarfism and microcephaly that are actually mutated in TOP3 or in RMI1 protein. And these individuals, again, have very characteristic look, as well as being very small. But interestingly, unlike Bloom syndrome, these individuals actually show mitochondrial dysfunction because this TRR complex, unlike BLM, actually translocates to the mitochondria as well. And indeed, most of these children actually will die very early in life because of mitochondrial abnormalities leading to cardiomyopathy. So what I'm going to talk about today is really two roles that we see for the BLM protein actually in mitosis, so during cell division. One is in the resolution of some structures that form in mitosis that we've worked on for many, many years, which are called ultrafine anaphase bridges, and I'll show you what we mean by that, which are UFBs, but also a new role that is currently unpublished, where we believe that BLM is very important for suppressing aneuploidy by acting as in a checkpoint role during mitosis. So, what are these anaphase bridges that I was mentioning? Now, it's been known for decades that when cells go through mitosis, when they go through the anaphase of mitosis, sometimes you can see a DNA thread, a bridge structure, using a DNA dye. But these are very, very rare, but they do exist. And of course, they're rather pathological structures. They're not going to be particularly useful structures during mitosis. But what wasn't known up until relatively recently is that there's a much more common DNA structure that arises during mitosis, but you can't see it because it doesn't stain with DNA dyes. And it only can be detected by immunofluorescent staining for proteins that are on this, so where this red bridge is here. And we call these bridges ultrafine anaphase bridges, and the reason why we work on them is the Bloom's complex of proteins is actually sitting on those bridges. It coats the bridge from end to end, which is also a rather curious phenomenon as to why you would want to do that. But there are a number of other proteins that sit on these bridge structures, and they're actually very common. They're actually not pathological structures. They arise in every normal mitosis. You don't need to perturb the cell cycle to see these. They're actually present in the early stages of almost every anaphase. But when you look down the microscope and see a cell going through anaphase, you don't realize there are DNA bridges there unless you stain for the appropriate proteins. And what is very important is that the absence of BLM, so if you take Bloom syndrome cells, for example, you get an increase in the frequency of these anaphase bridge structures, but also a delay in the resolution of these bridges. So instead of them being resolved very early during anaphase, they tend to sit for long periods of time well into late mitosis, normally when most of these bridge structures have already been resolved. They're actively resolved during anaphase and telophase. So, how do cells deal with these? If they don't get resolved, what happens if the cell gets into late mitosis and it's basically still got a kind of DNA tether that's sitting in between the separating DNA masses? How would it deal with this? Well, one thing that it actually does is have a checkpoint. It actually has a check on whether it's time to go through final cell division, cytokinesis. And that checkpoint is controlled by a DNA kinase, by a protein kinase, Aurora B, and it's called the abscission checkpoint. So abscission is the process, the sort of final cut of mitosis when the two daughter cells come apart. And that's determined by a checkpoint. And if there's no DNA structure, the cell can go through mitosis just fine and it can undergo cell division. But if there's a DNA bridge sitting between the two separating cells, shown in red here, And in particular, if that DNA is passing through a structure called the midbody, which is a sort of dense structure that you see in all cell division events, then the cell says, stop, we can't go through abscission, we can't finish mitosis. We have to stay at this point and hold the cell while we try and do something about this bridge structure. And so what you get in a situation like that is that these cells accumulate in the very, very late stages of mitosis. And I won't go through the data to tell you what we think BLM does. I'll just show you the model for how we think BLM is actually influencing this process through its ability to bind to these ultrafine DNA bridge structures. So we know BLM is actually itself very heavily post-translationally modified when cells enter into mitosis. So BLM is not just regulated during S phase, where it plays various roles, but it's also very heavily regulated in mitosis. And if a double-stranded piece of DNA is identified there, these ultrafine bridges, BLM binds to that structure. And what we've actually found is that what BLM is doing on those structures, particularly in late mitosis, is it's actually using its helicase function, and what helicases do is pull apart the two DNA strands of DNA. What BLM is doing is converting double-stranded DNA to single-stranded DNA. And that single-stranded DNA is bound by the main single-stranded DNA binding protein in human cells called replication protein A, or RPA. And actually, what then RPA does is actually do this checkpoint signaling role. And it's been known for many, many years that RPA is a critical factor in the S phase of the cell cycle for signaling DNA damage by coating single-stranded DNA. And what we believe is that this mitotic checkpoint is essentially identical to the S phase checkpoint. It's just, you might say, moonlighting at a different stage of the cell cycle. So what RPA does is it activates the so-called ATR-ATRIP kinase pathway, which leads to the phosphorylation of another kinase, CHEK1. And what CHEK1 does is then actually phosphorylate Aurora B, which is the mitotic kinase that's critical for this checkpoint. Aurora B, when it's phosphorylated, moves to this mid-body structure, this dense structure that you see in late mitosis, and that is what triggers abscission delay. So this signaling cascade using multiple kinases is actually the S-phase DNA damage or S-phase replication checkpoint, and it's just being used in an identical way in mitosis. But the way in which you create single-stranded DNA to activate that in mitosis is through unwinding of ultrafine anaphase bridges by a DNA helicase, which is BLM. And just to show one piece of data to show you that there is a clear abnormality in Bloom syndrome cells, here are some cells. This doesn't come from a Bloom's patient. This is a CRISPR knockout of it in a cancer cell line. But in wild-type human cells, you get the accumulation of phosphorylated Aurora B in the mid-body, or indeed in the mid-body arms, as they're called. But in BLM cells, that doesn't happen. You can't activate Aurora B in the same way that you can in wild-type cells. So the question then arises, if you can't, in a Bloom syndrome cell line, you can't activate the checkpoint, and yet you still have problems segregating your DNA properly, what are the consequences of that? There must be a consequence of that, because otherwise they wouldn't bother with the checkpoint, you might say. And the consequence of that, primarily, actually, is that the cell just gives up on mitosis. It basically aborts mitosis, and what happens in Bloom syndrome cells or BLM deficient cells, not in wild-type cells, is that you accumulate an enormous frequency of binucleated cells. The cell has two nuclei in one cell. And that's now very characteristic of Bloom cells. You get these binucleated cells, and we believe that that is because they don't have this mitotic checkpoint, and they can't control the timing of DNA separation in mitosis. And we know that this is actually a role for BLM that's directly in mitosis, and that's because we actually created a so-called Daggeron cell line. So a cell line where we can inducibly degrade the BLM protein. And we degrade it only in mitosis, and we get the same phenotype. So this is a role that doesn't require BLM to be inactive during S phase, for example. So now we've actually ended up with a situation where we can induce a very high frequency of binucleation, which is effectively tetraploidization. And that led us to hypothesize that this tetraploidization is probably a key driver of aneuploidy in Bloom syndrome cells. Bloom cells are actually notoriously aneuploid, and they are, of course, Bloom syndrome individuals, of course, cancer predisposed. And so our argument is that this could be a key driver of tumorigenesis in Bloom syndrome, because it's a way of actually getting a high frequency of aneuploidy quite straightforwardly, you might say. And the reason why we think that producing a binucleated or tetraploid cell is such a driver of aneuploidy is that following DNA replication in these cells, most of the time you get a highly chaotic first mitosis. You get multipolar spindles instead of a normal bipolar spindle. And then what happens is you just segregate a whole series of different aneuploid cells, most of which have a selective disadvantage, of course, compared to normal cells. But occasionally, some of these cells have characteristics that are actually selected for, and that's a very strong driver, we believe, of tumorigenesis. So I'll just summarize what I've said. BLM deficiency leads to excessive chromosomal instability, including aneuploidy. BLM deficient cells fail to generate these replication A protein coated ultrafine bridges in telophase, and hence they can't signal the presence of unresolved DNA to the abscission machinery. We think that abscission checkpoint deficiency generates tetraploidy at a high frequency in BLM deficient cells, which we think very likely contributes to cancer predisposition in individuals with Bloom syndrome. And these are the people who've done the work, and these are our funding agencies, and the two people highlighted with the yellow boxes, Rahul and Manika, two Indian postdocs, have been doing this work, and I thank you for your attention.
Thank you very much. Yeah, question?
Ian, wonderful talk. I was wondering, this general tumor spectrum that you see in Bloom, is that associated with an elevated level of then structural variants instead of single nucleotide variants that somatically accumulate?
Yeah, it is associated with structural variants, yeah. I'm not sure to what extent there's been extensive studies of single nucleotide variants, but certainly structural variants accumulate very substantially here.
Are there more questions? Yeah, all the way in the back.
My voice. Thank you very much for an interesting talk. So do you think it's a general role of the helicases, or it's specific to Bloom to have this effect on abscission?
As far as we're aware, the mitotic role of BLM is, you might say, unique to BLM. We have yet to identify related helicases on these DNA structures in mitosis. That doesn't mean to say they aren't playing a role in mitosis. But as things stand at the moment, this appears to be something that BLM does and not the other RECQ helicases, for example. There are indeed some other helicases on these bridge structures, but they aren't the RECQ helicases, the related ones. The FANChem helicase, for example, is on these bridges.
Thank you, Ian. Thank you to the speakers. It has been an excellent start for this morning session, so thank you very much.